Maybe we're going to record it. Okay, so in this first section of photosynthesis, what we want to introduce is how photosynthesis is able to take the energy that's found in light and help use that energy to transfer that into the chemical energy of sugars. Okay, so this kind of sets up the preliminary stages as far as where that's taking place at and how that's going to occur. And when we look at it, one of the areas that we really want to focus on, if we examine the graphic on the side, is we're going to look at where are these processes specifically taking place at. So we have the solar energy coming in, and obviously we think about our leaves as our area of primary photosynthesis, and then we'll take a look at the leaf structure, and we'll look at the cells that make up the internal leaf structure, and then we'll zoom into the chloroplast, and eventually we'll get down to the thylakoid and the structures of the thylakoid that are embedded inside that chloroplast. So a couple things that we want to look at as we introduce this idea. The structure that's the most important and distinguishing in terms of photosynthetic activity is the chloroplast. And once again, when we think about the concept of the endosymbiotic theory, which we addressed during the respiration chapter, the concept is that some of the early prokaryotes were invaded by other prokaryotic organisms. And we see the mitochondria as an example of one of those types of structures. And we look at the evidence that exists with the mitochondria. We know that the external membrane structure is very similar to how bacteria are organized. We know that mitochondria have DNA on their own. They have ribosomes. They have self-replicating capability. When we look at the chloroplast, we see a lot of those same characteristics. Okay, they have a membrane very similar to prokaryotes. They have ribosomes. They have their own genetic material. So this structure is another point of evidence that's used in the endosymbiotic theory explanation. And like I said, they're very similar and likely have evolved from some of the photosynthetic bacteria that existed in the early environment. Okay, so like I said, this is a key piece of evidence in the endosymbiotic theory, and that concept is definitely something that is addressed throughout the College Board exam. So you just want to make sure that you're familiar with that, and we'll examine it at a variety of different points throughout the course. Another thing to keep in mind, and it was mentioned in the preliminary write-up to this chapter, is that the early atmosphere didn't have oxygen. The early atmosphere was devoid of free O2, and it was the early photosynthetic organisms that emerged that started to run the process of photosynthesis and started to give off that byproduct of O2 that allowed us to accumulate and eventually build an environment that has oxygen in the atmosphere. Okay, so I think we need to start by looking really closely at where does this stuff take place at. Of course, it's occurring in the chloroplast. And when we think about the structures in a plant where photosynthesis is going to be primarily taking place, the leaf structures are going to be your major location. Okay, and typically when we look at plants during the heart of their growing season, the color that emerges is obviously green, and that green color is coming from a pigment known as chlorophyll. Okay, and that pigment's embedded within the chloroplast. And we're going to take a look a little bit today as to how that pigment plays such a critical role in allowing us to generate the energy that's necessary to build sugars. So what we find is that light energy that is coming down and being exposed to our particular leaf structures is going to be absorbed in the chloroplast by the chemicals of chlorophyll, as well as some of the other pigments, which are going to be referred to as accessory pigments. So we have more than just one pigment type. Chlorophyll A would be our what we consider our dominant pigment, but we're going to see that there are other pigments that are available as well. And what they're going to do is they're going to take that energy and they're going to drive the formation of the organic molecules, which primarily are going to be the sugar molecules that we're trying to develop through this process. Okay, so let's talk a little bit about what photosynthesis relies on. So, if we go into the chemical reaction of photosynthesis, okay, and we take a look at it, we can basically say that we're going to take more or less the products of photosynthesis, or of respiration, so we looked a lot in the previous section about taking glucose and oxygen and being able to release carbon dioxide and water. So photosynthesis on the surface looks like the reverse process of cellular respiration. Obviously, the internal workings of how all of this is accomplished are not exactly mirror images of each other, but the outcome overall does appear to be the reverse reaction. So what we're going to find is that the plants obviously are going to need a supply of both carbon dioxide and water, and we're going to see that the carbon dioxide is going to enter into our leaves through special openings, which are called stomata. Okay, so when we look at it, we'll show you that it's primarily on the underside of the leaf, the structural component of this, and the stomata is something that is able to be regulated in terms of how it's opened or closed. So it's a point where it's an opening to the internal portion of our leaf and the cells inside the leaf, and we're going to find that the things that are necessary to come in regards to carbon dioxide, they're going to enter in through that stomata. And we're also going to see that the waste product of oxygen is also going to be able to exit through the stomata. But another consequence of opening that up is that there's also the possibility of losing water through transpiration. So we're going to examine how different cells of, or I should say how different plants found in different environments, how they deal with that consequence of potentially losing water through the opening. And if you're in a very arid environment with a lot of evaporation, that could obviously create an extra stress on the system. All right, so we see that the CO2 is going to enter and the carbon dioxide is going to leave through the stomata. One thing that we've done before, obviously we're not physically here, is we've taken a leaf and we've put like clear fingernail polish on the underside of it. And you let it dry and you peel that off, and you can look at it under a microscope, and it allows you in many ways to see imaging of that structure that's underneath there. And you could potentially see the openings, which are the stomata. So what we find is that chloroplasts are found mainly in the cells of the mesophyll, which is going to be the interior tissue of the cell. So when you think about a leaf, if you kind of squeeze it, you got like a little bit of a sponginess to it. So the mesophyll layer of cells is that spongy interior portion, and that's where most of our chloroplasts are going to be. And we're going to find out that a mesophyll cell doesn't just have a single chloroplast. Much like cells didn't have just one mitochondria, we're going to find that these cells of the plants aren't going to have just a single chloroplast, but rather they're going to have accumulations of them, which might be 30 to 40 per cell type. So here's an image of what the cell structures look like within a typical leaf. And one of the things that we wanted to focus on is right here. So here's the underside, and there's the stomata, there's the opening. Now, the opening is regulated by the cells adjacent to it, which are guard cells. So those guard cells can be closed or they can be opened, and that's going to be, in many cases, a function of the environment. So we're going to see how that's going to operate. And then other things that are inside here, you're going to find the mesophyll cells. And like I said, these mesophyll cells, you might be able to see that there's this dark structure inside that. That would be your chloroplast. we know that the cells, or that the leaves tend to have sort of like a waxy surface to them. That's the cuticle, which in many ways helps protect it. So you got a cuticle on the top and the bottom of the leaves. You have your vascular tissue. Okay, the vascular tissue is going to help us move both nutrients as well as water throughout the leaf structure. We're going to talk a little bit later about the cells that line this vascular tissue, called the bundle sheath cells. Some of our different adaptations are going to utilize that in the process of making sugar. So it all just depends on where the cell, where this plant is most adapted to life at. Okay. So I like this particular slide just because it gives us a little bit of a progression in terms of where things are located at. So, we understand what a leaf looks like. We're zooming in on the leaf. And like I said, within that leaf structure, you have all of these mesophyll cells that populate the internal area of the leaf. And, you have your openings on the underside, which are going to regulate things like carbon dioxide and oxygen moving across there. The vein systems right here. These are the chloroplasts, which are embedded inside there. Inside the chloroplast, you have these thylakoids, okay, which we're going to take a look at, but they're stacked into these columns, which are referred to as grana, all right? And this is the heart of where photosynthesis is taking place. So you have multiple chloroplasts inside each of the mesophyll cells. You obviously have thousands of mesophyll cells within each leaf, and as a whole, we're able to go through there and build this sugar building capacity. So we see that the chlorophyll is in the membrane of a structure known as a thylakoid, okay, which are ultimately going to, if we look at it, the thylakoid kind of has a structure similar to what we saw with within our mitochondrion, but we don't have that heavily folded inner membrane. Okay, so if this is a thylakoid, they tend to stack up kind of like this within the structure of the chloroplast. So if I were showing you the overall chloroplast right here, I would have these stacks of these thylakoids embedded within the internal area of the chloroplast. Okay, so the thylakoid as a whole has a lipid bilayer. It has a double membrane system. And when we start talking about things like the photosystems, our photosystems, which are going to be photosystem number 2 and photosystem number 1, and there's going to be an electron transport chain. That's where it's going to be located at. It's going to be in the structure of that thylakoid. And what we're going to do is we're going to use the pigments, which are going to be found inside these photosystems, both photosystem 2 and 1, and we're going to be able to use the radiant energy, the solar energy, to drive energy production inside that chloroplast. Okay, so if we look at this here, we said that they could be stacked into grana, and the chloroplast also contain a dense fluid, which is going to be your stromal fluid, or the stroma. So, overall, if this is my chloroplast structure, and, within that chloroplast, which, like I said, in and of itself, the chloroplast has a membrane like this. We have the thylakoids stacked up like this. So we know that the thylakoids tend to stack into grana. So here's a stack. Here's an individual thylakoid, one of these things. And the liquid inside there is the stroma. Okay, so that's our general layout of what makes up a chloroplast. So much like when we did photosynthesis, we knew that the different areas of the structures were responsible for different processes. Same thing's going to be true when we get into photosynthesis. We're going to find out that in some cases, we're going to be able to make ATP, and in other cases, we're going to actually be able to use that to make sugar. Okay. All right. So this image right here, this is showing us a thylakoid zoomed in. And the connection that I want to make sure that we get is this looks very similar to what was going on in the mitochondria in terms of cellular respiration, electron transport chains. This electron transport chain is obviously specific for the process of photosynthesis. We're going to find that this is powered by the light energy. So we're going to show you how light energy can actually power the movement of these electrons to get them flowing. We're also going to see that those electrons are going to play a role in helping us push protons across the membrane. So much like what we saw in respiration, we build up an electrochemical gradient. We're getting a gradient of positive protons on the inside, these hydrogen ions, compared to what's outside of that thylakoid. We're going to see the enzyme ATP synthase play a critical role once again, because the hydrogen ions are going to be able to essentially go from high concentration to low concentration through that metabolic rotor of sorts to make ATP. And then the other thing that we're going to introduce is in the close family, in respiration, we had NAD being converted into NADH plus H. In the case of photosynthesis, they have a very close relative. NADP is going to be converted into NADPH. So an easy way to remember that is plants or photosynthesis has the one with the P. Okay? Respiration doesn't have the P on that. So if you're thinking photosynthesis or plants, think about the energy molecule that has the P inside of it. Okay. All right. So we'll use this as a little bit of a preview to kind of show us where we're going with the stages of photosynthesis. And there's two main stages. Okay. Now, depending on what source you look at, sometimes they call them the light reactions and the dark reactions. The light reactions are definitely going to be an acceptable name. The dark reactions are probably not super accurate, because even though the reaction technically is not dependent upon the light at that particular portion, as soon as the light stops, the reaction stops. So we're going to see that there's some other ways to define that. But the two areas, the two processes that we're going to focus on, we have the light-dependent reactions. That's when we're actually using the solar energy coming in from the sun. Okay, so the solar photons that are going to come into that system, we're going to see that they're going to be focusing in on those pigments within the thylakoid, and they're going to be generating things like ATP and NADPH, which means that there are going to be energy molecules and they're going to be electron-carrying molecules. The second portion is when we take in the carbon dioxide and we use the Calvin cycle to produce the sugars. Okay, so that would be our second portion of this overall system. So the light reactions are going to take place in the thylakoids, and the big things that we're going to see is that they're going to take in water. Okay, so the investment of water, the investment of light, and we're going to see some oxygen being released out of that situation. And we're going to see the redox reaction. So we're going to generate this molecule. And what does this molecule carry? It carries with it high energy electrons and protons. Okay, so we pick that up very much like what we did during the respiration process. And the other thing that we're going to do is we're going to generate ATP. So making sugar, if you think about it, we're making C6H12O6. Okay, that's an anabolic process. So if we went back to chapter 8 and we thought about, the energy associated with different types of reactions, and we said, all right, well, we're taking 6 CO2s and 6 waters, and we're going to make a glucose molecule, and we're going to release some oxygen, and obviously we're going to need sunlight. So if I were going to look at a general energy graph, so if I look at my change in free energy over time, we know that what we start with, my reactants right here, my products are going to have more energy in it. Okay, so in order for me to have a positive delta G, that means that I am absolutely going to be relying on energy coming in. So the energy of sunlight is going to be our initial source. And we're going to use that solar energy to generate a chemical energy, which is ATP, so that we can use that to eventually build up this high energy molecule, right? We know we can quantify this 686 kilocalories per mole of energy. How are we going to build that? We got to use an investment of light. We got to convert that light into chemical energy, such as ATP and NADPH. And then once we have all of those things in place, then we can actually execute the sugar production portion. Okay? All right, the Calvin cycle, named after Melvin Calvin, sometimes also referred to as the light independent reactions, that's happening in the stroma portion, okay? So remember, if our chloroplast looks like this, and we had the light reactions within these thylakoids that are stacked together, the inner liquid portion, which would be like this area right here, this is the stroma. And within the stroma, we are able to use the products of the light reactions. So this area, this gray area through here, is going to drive the cell through the Calvin cycle. And what we find is the Calvin cycle is going to use something called carbon fixation. Okay, so when you take carbon and you pull it out of the atmosphere, such as CO2, and you lock it up into organic molecules, that's the process of carbon fixation. Okay, and when we take a look at from an environmental perspective, in regards to how our carbon is being stored and how our carbon is being released, if you look at certain components, such as fossil fuels, for example, those were obviously molecules that were stored from living organisms that were compacted under the earth for tens of thousands of years in order to generate the molecules that we get in things such as our carbon-based fossil fuels. Okay, so that's our general introduction into photosynthesis. So in our next section, we'll get into a little bit more detail about the light reactions.